So I think some of the limitations that exist right now within the CAR T world is actually within solid tumors. I think that there's a lot of CAR Ts that are doing wonders within the hematologic malignancies, but translating that new modality, which is so promising, from the liquid tumors into the solid tumors has been met with some challenges. I think some of these challenges involve having targets. We have found targets with NY-ESO, we've found targets with MAGE-A4, MAGE-A10, but translating these across from identification of patients to translation and to clinical trials has been a challenge, mainly because some of the rarities of finding patients that actually match these criteria. The adaptimmune SPEAR T cell is a new technology that's been in clinical trial for a number of years now. It begins by taking T cells out of a patient and then reprograms them to go after targets. You get both an intracellular and an extracellular ability to identify targets, and then you put these modified T cells back into a patient with a conditioning regimen so that these cells can establish themselves and propagate, and then attack the cancer. And since this is based on a patient's own immune system, outside of the window where you're actually getting chemotherapy to establish the niche to put these cells back into, the toxicities have been incredibly low outside of the actual initial therapy period. And these cells, because they propagate, are actually useful because you continue to treat your cancer without getting treatment side effects like you would every 3 weeks with a chemotherapy regimen persistently for as long as the T cells persist in the body. So the SPEARHEAD-1 trial is a confirmatory and registration Phase II trial looking for both synovial sarcoma and myxoid round cell liposarcoma patients who are HLA-A*02 and who express the testicular antigen MAGE-A4. And so this is a trial where, based on the Phase I data, where the synovial sarcoma data was not only had a response rate, but was durable with 4 patients going greater than a year in terms of response. The SPEARHEAD is the confirmatory trial within the sarcoma patients for FDA registration, once again using a patient's own T cells, modifying them and going after the target. In synovial sarcoma and myxoid round cell sarcoma, about 40% of every patient screened is expected to be HLA-A*02, with the majority of those that are HLA-A*02 thought to also be positive for MAGE-A4. So a worldwide effort right now is underway to find patients who are HLA-A*02 and MAGE-A4 positive to put them in this study. What is interesting is that being both HLA-A*02 and MAGE-A4 positive isn't limited to just a sarcoma population. There's a large screening effort underway to identify patients with lung cancer, head and neck cancer, ovarian cancer, bladder cancer, melanoma, or any other tumor where there's thought to be both HLA-2 and MAGE-A4 positivity. And so because of that, including urothelial patients, there really is a lot of solid tumors where there's a low percentage of patients that would qualify for a therapy like this. Within sarcoma, if and when, I'm very hopeful about this, that the SPEAR T cell for synovial sarcoma and myxoid round cell gets approved, I think this is going to dramatically change our approach and our impact of patients with synovial sarcoma and myxoid round cell. I think that the data that exists right now is in heavily pretreated patients. I think we'll be moving our approach up to screening very early and treating with something like this very early, especially in the metastatic setting. And so I think it's going to really change how we approach patients that are HLA-A*02 that express this antigen. And so as patients come in, I would expect them to be screened very early and then probably treated as quickly as we can get them access to the therapy. So I think that this will be practice changing and practice transformative, because I'd like to see if this works better as patients are treated in earlier lines of therapy. As with most therapies, the earlier you use it, the more likely it is to work. And so I'm hoping as we push this further up in line that we will actually see even better and more prolonged responses.